Immunology Insights, Basics and Beyond, Chapter 58, Monoclonal Antibodies in Cancer Treatment. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we paid tribute to Charles Richet, the scientist who first recognized and described anaphylaxis and how careful observation can redefine medicine. Today, we changed perspective once again. After examining immune overreaction in allergy and asthma and previously laying groundwork in tumor and cancer immunology, we now move fully into immune-based cancer therapies. Cancer is particularly suited for immunotherapy because immune cells already sense abnormal transformation. Yet tumors exploit regulatory limits to survive. One of the earliest and most powerful clinical tools that emerged from this understanding is the monoclonal antibody, or mAb, engineered proteins that can tag, block, or directly eliminate cancer cells or even recruit the immune system to finish the job. This chapter begins our next deep dive series into cancer therapy, starting with mAbs, the blueprint for many modern immunomedicines. So let's talk science. What are monoclonal antibodies? Monoclonal antibodies are lab-made immune proteins from a single B-cell clone, so all molecules bind the same epitope on one antigen. This precision makes them predictable drugs. One mAb equals one target that equals one main mechanism. Polyclonal antibodies, so the normal serum response, are mixed populations with variable specificity. Because therapy needs a defined, predictable molecule, almost all antibody drugs are monoclonal. You can recognize them by the generic name ending mAb or mAb. For example, rituximab, trastuzumab, or pembrolizumab. Well, how are monoclonal antibodies made? Originally, mAbs came from hybridoma technology. B cells from immunized mice that make the desired antibodies are fused with immortal myeloma cells. The resulting hybridoma secretes one antibody indefinitely. Today, modern methods such as phage display and transgenic mice with human immunoglobulin genes generate human or near-human antibodies. Typical steps involve: first, choose a cancer-relevant antigen, for example CD20 or B cells or HER2 on some breast cancers. Second, immunize or screen libraries and select high-affinity binders, clone and humanize the antibody genes. Third, express the final antibody in mammalian cells, often Chinese hamster ovarian cells or shorter CHO cells, and purify it as a drug. In cancer therapy, mAbs recognize tumor-specific or tumor-associated antigens, block growth and survival signals, recruit immune effector functions such as ADCC and complement, or deliver toxic payloads or radiation to kill the tumor. Now let's have a look at antibody engineering, from mouse to human. First-generation antibodies were fully murine and often triggered strong anti-drug immune responses. Engineering therefore moved through chimeric antibodies, which were around 65% human and still contained a murine variable plus a human constant region. One known example is rituximab. Then humanized antibodies, which were already 90 to 95% human, and only the complementary determining regions were murine, and now fully human antibodies, created via transgenic mice or phage display to minimize immunogenicity and improve pharmacokinetics. What are the main mechanisms how monoclonal antibodies can fight cancer? First, blocking growth and survival signals. Many tumors depend on overreactive receptors that constantly send grow and divide signals. mAbs block these receptors and interrupt signaling. Trastuzumab against HER2 in some breast cancers and gastric cancers, and cetuximab or panitumumab against EGFR in colorectal and head and neck cancers are classic examples. These agents rarely kill cells alone, but deprive them of survival signals and make them more sensitive to chemotherapy, radiation, or immune attack. Second, flagging cancer cells, so ADCC and CDC. When a monoclonal antibody coats a tumor cell, its Fc regions becomes a flag for immune defense. In ADCC, so antibody-dependent cellular cytotoxicity, Fc regions bind Fc gamma receptors such as CD16 on NK cells and macrophages, which then release perforin and granzymes. In CDC, so complement-dependent cytotoxicity, antibody binding activates complement and forms membrane attack complexes that lyse the cell. Rituximab, an anti-CD20 antibody for B-cell lymphomas and leukemias, uses both ADCC and CDC mechanisms to deplete malignant B cells. Fc engineering, such as R-fucosylated IgG, can increase Fc gamma receptor binding and boost ADCC in newer antibodies. Third, checkpoint blockade, releasing the brakes. Some monoclonal antibodies target immune checkpoints rather than the tumor cells. Anti-CTLA4, for example ipilimumab, sustains early T-cell activation. Anti-PD-1, for example nivolumab or pembrolizumab, blocks PD-1 on T cells. Anti-PD-1 ligand or anti-PDL1, for example atezolizumab or durvalumab, binds PDL1 on tumor or stromal cells. By interrupting PD-1, PDL1 or CTLA4 B7 interactions, these drugs release the brakes on cytotoxic T cells so they can recognize and kill tumor cells. We discussed checkpoint inhibition in one of our previous chapters more extensively. Fourth, delivering target toxins or radiation. Antibodies can act as delivery vehicles. In antibody-drug conjugates, a monoclonal antibody is linked to a highly potent chemotherapy via a cleavable linker. After binding and internalization, the toxin is released inside the tumor cell. Brentuximab vedotin, that targets CD30, and the trastuzumab emtansine, that target TDM1 and HER2, are key examples. Radioimmunoconjugates such as ibrutinib tioxetan, that targets CD20 with yttrium-90, deliver focused radiation, and immunotoxins fuse antibodies or fragments to bacterial or plant toxins that kill cells after internalization. And fifth, modifying the tumor microenvironment. The tumor microenvironment, or TME, often protects cancer. Some monoclonal antibodies reshape this ecosystem. Anti-VEGF antibodies such as bevacizumab block angiogenesis, starving tumors and helping normalize abnormal vessels. Anti-CD47 antibodies block a "don't eat me" signal to macrophages via SIRP-alpha, restoring phagocytosis of tumor cells. Because red blood cells also express CD47, dose and schedule must be carefully managed. These strategies show that monoclonal antibodies can influence both tumor cells and their supportive environment. Now let's look at combination therapies, where monoclonal antibodies serve as backbone. In practice, mAbs are mostly used in combinations. With chemotherapy, rituximab plus CHOP in B-cell lymphomas is a classic example. Chemotherapy debulks, the antibody adds targeted immune-mediated killing. With radiotherapy, mAbs can sensitize tumors or be given as radioimmunoconjugates. With checkpoint inhibitors, growth blocking or ADC-type antibodies increase antigen release as tumors die, and checkpoint blockade lets T cells recognize these antigens. With cell-based therapies, bispecifics and BiTEs, such as blinatumab, a conjugate of CD19 and CD3, physically connect T cells to B cell tumors and trigger without full CAR T cell manufacturing. We also have to look at biomarkers and resistance, as not every tumor or patient benefits from the same antibody. Needed key biomarkers include antigen expression, for example HER2 for trastuzumab, CD20 for rituximab, or BCMA in some myelomas, tumor mutational burden, as high tumor mutational burden often means more neoantigens and better checkpoint responses, Fc gamma receptor polymorphism that affect ADCC strength, and the degree of immune infiltrate. So hot T-cell rich versus cold immune poor tumors. Together, these features support precision oncology, matching antibodies to the biology of each tumor. Even with good biomarkers, resistance can emerge. Tumor cells may lose or mutate the target antigen. For example, CD19 negative relapse after an anti-CD19 therapy. The microenvironment may stay immunosuppressive, so high IL-10, TGF-beta, Tregs, myeloid-derived suppressor cells, or alternative signaling pathways may compensate when one receptor, such as HER2, is blocked. In solid tumors, additional barriers include dense stroma and abnormal vasculature that limit penetration, hypoxia and acidity that impair immune function, and antigen heterogeneity within one lesion. So, let's look at future directions and how monoclonal antibodies can get smarter. New generations of mAbs aim to extend success in blood cancers to complex solid tumors. Core ideas include bispecific and multispecific antibodies that link tumor cells to T cells, as in blinatumomab and next-generation CD3-based designs, advanced Fc engineering to fine-tune effector functions and half-life, and immunocytokines that deliver cytokines such as IL-2 or IL-12 directly to tumors. Smaller formats such as single-chain FVs or scFVs or nanobodies may penetrate dense tissue better, while antibodies against stromal markers like fibroblast activation protein or FAP or abnormal vasculature can remodel the tumor bed or tumor microenvironment and improve immune and drug access. Monoclonal antibodies are also increasingly integrated within engineering cell therapies. For example, allogeneic CAR-T or gamma delta T cell therapies, where they can block inhibitory pathways or clear competing cells. Experimental mRNA encoded antibodies use lipid nanoparticles to deliver mRNA so that the patient's own cells temporarily produce therapeutic antibodies in vivo, potentially simplifying manufacturing and dosing. To conclude, monoclonal antibodies have fundamentally reshaped cancer therapy. They have transformed many hematologic malignancies, where targets like CD20, CD19, CD38, and BCMA are well defined and uniformly expressed, from uniformly fatal diseases into conditions with high remission rates. In solid tumors, progress has been slower but steadily improving, driven by better target selection, more sophisticated engineering, and smarter combination strategies. Their two strengths lies within their versatility. Monoclonal antibodies can block oncogenic signals, mark tumor cells for immune destruction, deliver cytotoxic payloads, reshape the tumor microenvironment, and modulate immune checkpoints, all with molecular specificity. In our next chapter, we will take the story further with next-generation checkpoint therapies and combination immunotherapy, exploring how refined control over immune activation paired with tools like monoclonal antibodies may bring us closer to durable and widespread cancer control. Before closing, I have a small request. If you enjoyed my content and want to hear more, please like and subscribe. It's a small step for you, but it helps me a lot. So thank you for listening and see you next time.